Welcome to this new video. We will be exploring the principles, techniques, and applications of protein separation in biochemistry and molecular biology. Proteins are like snowflakes. Each one is unique. We separate them based on their physical and chemical properties like size, charge, and affinity. Understanding this principle is crucial for selecting the appropriate separation technique. Let's start with gel electrophoresis, especially SDS-PAGE. Imagine running a race but on a gel track. SDS gives proteins a uniform negative charge so they run towards the positive end with the smaller proteins running faster. It's like a protein marathon. Next we have isoelectric focusing or IEF. This technique separates proteins based on their isoelectric points or pi. Proteins migrate through a pH gradient until they reach the pH that corresponds to their pi, where they have no net charge and stops moving. Now, let's talk about chromatography. This versatile technique separates proteins based on their different properties. We will look at three common types: affinity chromatography, ion exchange chromatography, and size exclusion chromatography. Affinity chromatography. This method uses specific interactions between a protein and a ligand attached to a stationary phase. It's highly specific and effective for purifying proteins from complex mixtures. Next up is ion exchange chromatography. This technique separates proteins based on their net charge. Proteins are passed through a column containing charged resins, and those with opposite charges bind to the resin and are eluted by changing the salt concentration. Last but not the least, size exclusion chromatography, also known as gel filtration, separates proteins based on their size. Larger molecules can pass through the column faster, while smaller molecules take a longer path through the porous beads. Protein separation techniques are widely used in various applications, including diagnostic testing, protein purification for research, and the development of biopharmaceuticals. These methods are fundamental for many areas of research and industry.